So it's my absolute privilege to introduce our EC member and a featured speaker, a man who treats the disease not just with medicines, but as a person. He's a doctor, Raghunadharao sir, who's a professor of medical oncology at MNJ Institute of Oncology and Regional Cancer Center. With over 3 decades of his dedicated service, sir has been guiding force in cancer research, clinical trials, and cancer prevention programs. So, beyond this credential, what stands out most is his commitment to advancing patient care and nurturing next generation of clinicians and researchers. And sir has been into service thing, like he has also sponsored many students even from Alphaba Academy for to taking a lot of workshops. Even for the past workshop also, sir has sponsored for many students, and he's actively into helping people to overcome various things. And he's also a recipient of Dr. BC Roy National Award. So let's join together and hit the emojis down there, and let's welcome sir to present on this Nobel Prize on Medicine and his views on this T-Rex cells in immunotherapy. Over to you, sir.
So I simply put the title as the T-Rex story, so that I first will take you through the story. Um, if you learn it like a story, you are more likely to remember it. And that's the reason why I have put it as the T-Rex story. And this is a statement from Professor Krishnamurthy, the son of Dr. Muthulakshmi Reddy, who founded the Cancer Institute in Adyar. So today's research is tomorrow's treatment. And what we are going to talk today is perhaps something which holds this statement very true. He made this statement when we were students. I was a student there in 87 to 89. And this is actually the legend beneath his photograph at the Cancer Institute now. So today's research is tomorrow's treatment. So most of you are from basic sciences. So you can be 100% sure that this is what is likely to be the truth. So most of us know this. There is a Nobel Prize for Physiology or Medicine, and it was awarded to Mary Brunkow and Fred Ramsdell and Shimon Sakaguchi. And they answered a very simple question: why do not our bodies destroy themselves? So the gist of what the Nobel Committee had said is they discovered the body's secret security guards. They are special cells that constantly patrol your body, making sure your defense system does not accidentally destroy you. So let us see how this story unfolds. So there are some security guards. They also seem to be dressed like anybody else, like rest of the defense. So you are trying to seek out somebody who is in plain clothes. And how did they unravel this mystery? Let us first hear. So in the 80s, when I was still a student, I actually used to work with Professor Shobha Sehgal at PGI in Chandigarh. She was very deeply interested in subclassification of T and B lymphocytes, especially the T lymphocytes. And I used to help her get the lymph nodes out of which we would tease out. In fact, I used to have a small coffee filter, a tea filter, in which I used to tease out the cells and then make a suspension of them to know whether T or B cell subsets. And we all thought that the puzzle was already, they said there are T cells, there are B cells, And they said there are some T cell subclasses, and we closed the chapter there. And that's exactly what I used to do when I was a student. So we believed that everything started from the thymus, which was a training camp for immune cells. So the immune cells can be from the bone marrow, 99.9% of the time. And then they go to the thymus, and the thymus has some epithelial cells where the thymus actually recruits cells and then destroys some others. So the scientists believe that rogue soldiers were eliminated during the boot camp in the thymus through a process called central tolerance. Others also did believe that there is another layer of protection with the cells, which they called suppressor cells. But the evidence fell apart, and the entire hypothesis was rejected in the 1980s because some experiments could not be duplicated and were found to be actually false claims. So everybody forgot about this, and they felt it is too toxic. Let there be B cells and T cells, that should be enough for us. Let us abandon the field. So they abandoned. There was one person, Shimon Sakaguchi, at Nagoya in Japan, who refused to give up. And let us see why he refused to give up. What actually kept him going? Let us see why he did that. So he did a bizarre experiment. He removed the thymus from a newborn mouse. That means as soon as it was born from its mother, he did a small incision. The thymus was just beneath the breastbone, and he just removed it, expecting that they have a weak immune system. So he removed it on the first day they were born, second day, the third day, the fourth day, fifth day, sixth day, etc. So, but when the operation took place 3 days after the mice were born, something very shocking happened. And the immune system did not actually go into an overdrive or run amok. It did not attack anything inside or everything inside. But very strangely, it did develop a range of autoimmune diseases. So it looked like the mice were actually destroying themselves. So we are led to believe that if you have an athymic mouse, that means a mouse which is born without a thymus, there is no defense system in its body, and everything will invade it. But the difference in what we know as athymic mice and the experiment which Shimon did is the difference in the time after which the thymus was removed from the body. So if you allowed the thymus to function for at least 3 days after the mice were born and then removed the thymus, the body started, the so-called T lymphocytes started attacking their own body parts, and they developed a large number of autoimmune disorders. So literally they were eating themselves up. So to him, this did not make sense. If the thymus was the only thing preventing a self-attack, why would removing it make things worse? So if you allowed the thymus to function for some time and then removed it, for example, let us say in a human being, you won't have a thymus once you cross between 9 and 14 years. The thymus simply atrophies. Unless there is a tumor in it which causes myasthenia gravis, which is again an autoimmune disorder, or a thymoma, which can be benign or malignant. No other diseases actually attack the thymus. It just atrophies. If you do autopsies in adult human beings, there is no thymus at all. It is just a mass of fibrous tissue. So if the thymus prevented self-attack, was the only thing which was preventing self-attack, why would removing it make things worse? So he wanted to test this wild idea. So he did something very strange. So he took healthy immune cells. So he did a phycal hyphae preparation, collected the buffy coat, and from normal mice and inject them into the mice without the thymus. So here you have a set of mice whose thymus was removed, a set of normal mice from whom he obtained the white blood cells. In fact, he did not separate them as T and B cells, and then injected them into the mice without a thymus. And suddenly, he realized the autoimmune disorder disappeared. So there is no thymus in the body. The body is attacking itself. But when you transplanted or injected immune cells from normal mice into the athymic mice, the autoimmune disorder abated. In fact, it disappeared. So these cells were like peacekeepers calming down the aggressive soldiers. So this set him into a thought process. So he wanted to know that without the thymus, where did these mystery gods come from? So he knew the mystery gods came from those mice which had a normal thymus. So he was convinced that the immune system must have some form of a security guard that calms down other cells and keeps them in check. So how do you spot this undercover agent in the crowd? So how do you know which of these cells circulating in the athymic mice or in the normal mice was actually this so-called security guard, which is preventing the body from destroying itself? Well, he did experimentation for over almost a decade. In fact, he did it over 11 years. And the protective cells were still wearing the same CD4 or helper T cell badges, but they were behaving completely differently from normal helper cells. And to his misfortune, he found that they are in extraordinarily small quantity or number given the weight of a mouse. So he then made a conclusion which was quite revolutionary, that there must be different types of cells which wear the same badge. So although they are wearing the same badge, they functionally appear to be different. This was his conclusion. So he then published this paper out, which arose from his department. Breakdown of a single mechanism of self-tolerance causes various autoimmune disorders. If you see from this paper, which was published in the Journal of Immunology in 1995, it's actually an embryonic science and technology department. You see read it where he is from: Precursory Research for Embryonic Science and Technology, Research and Development Corporation of Japan, and the Institute for Physical and Chemical Research, and of course the Department of Medicine and the Department of Dermatology from the Tsukuba University School of Medicine in Tsukuba in Japan. And he said, taken together, these results indicate that CD4 positive, CD25 positive, CD25 is nothing but an interleukin 2 receptor alpha chain molecule. Positive cells contribute to maintaining self-tolerance by downregulating immune responses to self and non-self antigens in an antigen non-specific manner. So this is what his conclusion was. Okay, so he made some presumptions as well, but the sum and substance was, yes, they did wear the CD4 badge, but additionally they expressed IL-2 receptor alpha chains, which he called them clustered of differentiation 25. So Sakaguchi defined a new class of T cells, which he showed that the T cells with CD25 on their surface protect against an autoimmune disease through this experiment in mice that lacked them. So if we injected the CD4 bearing T cells into the mice, but removed all the cells with CD25, the mice developed serious autoimmune diseases. If we added the CD25 bearing cells, the mice remained healthy. So the importance is here that it is not just CD4. The CD4, of course, is the helper T cell as we call it now. So without the CD25, it goes on to start destroying everything in the body, and there is no control over what it can destroy. But to this mass of CD4 positive cells, if you added a few, just a few, mind you, just a few, I believe he said 1 in 1,000 to 1 in a million CD25 positive cells, the mouse remains healthy. So it is like something like it has a supervisory or a calming security guard like effect in the area. It is like putting a poster, although nothing may be working, warning you are under CD camera surveillance, okay, video surveillance. So that statement is enough to keep people in check. Even if that camera is not functional, if people read that, they feel that yes, somebody is watching over you. So the unique signature is these special peacekeeping cells were not just CD4 positive badges, but also a second one called CD25. At that time, when we published this paper, many scientists were skeptical, they wanted more proof. We will stop his story now and move across to the Pacific and then see what happened in the Americas. Most of you are very familiar with the hydrogen bomb, which was dropped on Japan and destroyed in Hiroshima and Nagasaki, that arose from a project in the 90s, 40s called the Manhattan Project. Oppenheimer, most of you may have seen the movie Oppenheimer, and they were studying what radiation does to living things. Well, if you are going to drop it on human beings, you might as well study and see what happens. They did study after they dropped the bomb amongst the survivors. You know how much, what was your age at which you got exposed to the radiation? That resulted in a host of other diseases. Well, acute radiation will kill you, not actually through a marrow depletion, but it actually kills you through a neural shock. So the brain just fries up, bheja fry, and then you die. But if you had used laboratory mice, something strange kept happening. Mark my words, male mice were being born with scaly, flaky skin, something like our dandruff, or if you want, massive psoriasis, massively swollen organs, and they also died within weeks. Very strangely, they kept this breeding these mice over the decades. 1940, 1950s, 60s, 70s, 80s, they kept on breeding these mice. So they call this strain of mice which were exposed to radiation and were all male, and they call this strain as scurfy. Scurfy is simply a name for dandruff. Okay, so they were scrofulous mice, nothing more than that. But nobody ever understood what was killing them. In the 1990s, genetic tools became sharper. Most of your seniors or faculty here knew about this, and they knew that scurfy mice organs were being attacked by their own immune cells. So if a male progeny was born to a pregnant mouse which was exposed to radiation, the those which carried the female phenotype escaped, but those which carried the male phenotype were attacked by their own immune cells and the organs were destroyed. So the tissues are being destroyed by inside, and for some reason this scurfy mutation appeared to cause a rebellion in the immune system. So the soldiers actually turned against their own fortress. So this intrigued these two people. The dying mice were telling something very important. If one could understand the molecular mechanism of the underlying mice disease, you can gain decisive insights in how autoimmune diseases arise in humans. So, Dr. Bunklow and Ramsdell, who were actually working at that time in a biotechnology firm or a molecular biology firm, which was looking for answers to autoimmune disorders, decided to hunt down this scurvy mutation, find the exact piece of broken DNA causing all this chaos. Remember, there was no NGS at that time. Okay. Remember, they still had to work with the same old southern blotting and same old PCR electrophoresis. So they knew that there was something wrong on the X chromosome. The male mice carried only one X chromosome, female carried two, and so the male which carried the single X chromosome suffered. So they focused their attention on the X chromosome in the scrofulous male mice. It has 170 pieces of genetic code, and they wanted to find out one broken piece in all that information. It seemed impossible, but they were determined, and they narrowed their search down to about 500,000 pieces of the code. So they painstakingly worked for checking every potential gene in year after year, gene after gene, month after month. They started searching for it. So on the 20th attempt and final gene, they could shout bingo. Okay. So after several years of dedicated work, they finally found the scurvy mutation. It belonged to a family of genes that act like master switches, turning other genes on and off and controlling how cells develop. Dr. Mary and Dr. Fred named the new gene FOXP. It's actually a fork at that fork head gene, and they named it as FOXP3. Actually, they gave it a name themselves. At the same time, the hunch led to a small human connection. There is a rare disease in boys. I told you it affects boys or male only, called IPEX. I'll come back to what IPEX is. Like in the scurvy mutation, it is linked to the X chromosome. Boys with IPEX suffer as their immune system attacks their own bodies and they die in infancy. Just like the mice, it looks like the same thing is conserved and happens in human boys also, the male issues also. They found the human version of the FOXP3 gene in a genetic database and helped by pediatricians from across the world. I told you it's a very rare disease. They collected samples from boys affected with IPEX. So they actually collected all the, whatever blood they could and analyzed. The hunch proved correct. Boys had the same harmful mutation in the FOXP3 gene. So here is the IPEX syndrome. Here is a child, young boy with the IPEX syndrome. Below that, I have shown you the baby. I hope you can see the baby here inside. This is the baby. This is the baby's chest. This is the baby's abdomen. The abdominal organs are swollen. This is the liver. This is, of course, the lower jaw. That is the cervical spine and the dorsal spine. And this is the head and face. That's the brain. What is interesting is not all this. Look at the material outside. This should have been crystal clear and echoic. That means there should not have been a signal here at all. Okay, that means it should have been as black as what we see in this part of the brain. So it should have been absolutely black. But this is speckled, and as if there is a muddy, murky amniotic fluid. The muddy, murky amniotic fluid occurs because the child is shedding all the cells on his skin into the amniotic fluid. I hope you understood. So that is the reason why they are floating around in the amniotic fluid. And if you have any idea that this is the murky fluid, you can actually sample this and get to identify the FOXP3 gene mutation. IPEX stands for immune dysregulation, polyendocrinopathy. Most children are born with diabetes at birth. Enteropathy, they have severe diarrhea, and of course it was X-linked, as I told you. Just like in the mice from the Manhattan experiment, we know that the IPEX syndrome also is a similar syndrome which occurs in human beings. And this is was actually published as a letter in Nature Genetics, and disruption of a new fork-head winged helix protein called scurfin. They named it the protein scurfin, results in the fatal lymphoproliferative disorder of this Scrophulus mouse. They also identified this is the gene, and they mapped it very comfortably, and they identified where that mutation is located. It's a frame shift resulting from the insertion, which leads to a truncated gene product lacking the carboxy terminal of the fork-head domain. So there are pretty important genes upstream, so which are important, and this is how they published their work in Nature Genetics in 2001. So this is what she actually said. We identified a gene defective in Scrophulus mice by combining high-resolution genetic and physical mapping with large-scale sequence analysis. The protein encoded by this gene, which she called it, she gave the designation FOXP3, is a new member of the fork-head winged helix family of transcriptional regulators and is highly conserved in humans. In the Scrophulus mice, a frameshift mutation results in a product lacking the forehead domain. So if the forehead domain is lost or the forehead domain has a mutation, it demonstrated a protein product called FOXP3 or scurfin we can call it, is essential for normal immune homeostasis. So this was the conclusion from the paper. So this is what the recent Nobel committee had given. Here is a scurfy mouse, and that is the X and Y genes. Of course, I told you it occurs only in the male mouse. The Y gene is completely normal, but the X gene is what has the mutation. The scurfy mutation causes a mutiny in the immune system, and both of them succeeded in narrowing down the area of the mutation and located it in the FOXP3 gene, which turned out to be decisive in the development of the regulatory T cells. So this is the important part of this part of work here across the Pacific. So we need to connect the dots. What does the FOXP3 do? And the puzzle came together 2 years after she published, both of them published this paper. Shimon Sakaguchi and other researchers convincingly proved that the FOXP3 gene controls the development of regulatory T cells. One identified the regulatory T cells, the other team identified the FOXP3 gene. And FOXP3 is the instruction manual for creating the security guards. Without it, the body cannot make these peacekeeping cells. Without the peacekeepers, the immune cells, when soldiers run while attacking everything in sight, and the regulatory T cells prevent other cells from mistakenly attacking the body's own tissue. So without the Treg cells, the body will start attacking itself. And this process, now to differentiate it from the so-called boot camp in the thymus, is called peripheral immune tolerance. Tregs also ensure that the immune system calms down once it has eliminated an invader. There is a huge proliferation of T lymphocytes when you have something invading your body, any pathogen invading your body. And once the need for the expansion of the T helper cell is over, you need to calm it down and bring it down to the normal non-diseased state. So that is once the invader is eliminated. So this is a graphical representation of how the Tregs with CD25 are actually formed. I'm not sure whether it has come out well. I should have broken it into 3. I have put them together. In panel A, you can see the FOXP3, and once that is expressed, then of course, if there is no cytokine stimulation, then there is a negative selection and the Treg cell spontaneously is eliminated. So there must be a cytokine stimulation for it to be sustained and for it to sustain itself in the body. So that is important. This is of course a thymic epithelial cell. I told you the thymic epithelial cells actually educate the T cells of what is self and what is not self, and then they regulate what can be released into the circulation and what cannot be released into the circulation. So most important part is that is functional in the pediatric age group up to the adolescence. Beyond that, of course, it is self-regulated by the body. So there is no more education. You have completed your schooling, you have become an adult, so continue throughout the life of the adult. And you don't need the school, go back to the school. None of you go back to the school, I presume. So also the educated T lymphocyte doesn't go back to the school. It remains in the body as a memory cell forever. So let us say a T cell has slipped through the test in the thymus and it reacts with a fragment of one's own body's protein. So let us think that this is a rogue T cell. Regulatory T cells will discover that attack is a mistake and calm it down. So that is the T regulatory cell, and it intervenes by patrolling and keeping the T cell from attacking a self cell. So this is the self cell, and the self cell is preventing. So unless you prevent that, you automatically get an autoimmune disease. Most of us think that autoimmune diseases occur only in women. No, it can occur in men and women as well. And every single organ in the body is prone for an autoimmune disease. So nothing very strange about it. A little bit about Tregs in health and disease. They form only 2 to 5% of adult peripheral blood CD4 positive cell repertoire. Majority of the immune cells function to promote inflammation. They are pro-inflammatory because you need to fight pathogen, you need to fight cancer. So you need to create a milieu where you can attract as many cells as possible to destroy the whatever is harming the body. So the Treg cells keep the immunity in check to maintain a homeostasis and prevent a pathology from occurring. So dysregulation of Treg cell function is linked to many diseases. And so if there's a failure of these cells to contain the antigen specific effector T cells, it will lead to an autoimmune disease, allergies like our urticaria, etc., and various other hyperinflammatory diseases. If you do a transplant, Treg cells prevent the alloreactive T cells from attacking the foreign tissue, because the cells which come from a donor find this body, the body into which they have been transplanted, as foreign. So they will first attack the liver and destroy it. If the liver is not functional, you are gone. Then they will attack the intestinal epithelium. It comes out like tubes. In fact, it actually comes out like tubes. Then of course they destroy the skin. So your eyelashes fall off, the eyebrows fall off. Typically eyelashes won't fall off even after chemotherapy, but eyelashes also fall off, and the skin peels off in layers. So if you want to prevent that, you need Treg cells. In contrast, tumors enhance Treg cell response. So actually the tumor coats itself with a layer of Treg cells. Very interesting. So the tumor seems to attract Treg cells around it so that it is protected from being attacked by the other effector T cells in the body. So that's we are still not sure how the Treg cell is actually attracted to the cancer, and then how it prevents the effector cells from attacking it. So you need to have a balance of the effector cells versus the Treg cells. That is critical for human health. Of course, the same it holds good for the mice as well. So tumors are very clever. They attract a large number of Tregs or security guards to surround themselves like a protective wall. Okay, much like our politicians, or what we would say, those who were on the other side of the law now coming on to this side of the law. So they protect themselves with the same security guards. one who was arresting them in the past and putting them in a cell, now is protecting them out as a politician. Same thing happens. So T-Rex cells unfortunately do the same thing. The guard seems to tell the immune system there is nothing to see here, move along. So if you can strip a tumor of the Tregs, they will become accessible to immune destruction. So this is what I have given, how they suppress Tregs, how we can suppress Tregs. So many things seem to happen simultaneously. It is true that at different points of time or at different phases of a tumor, different things can happen. Of course, it need not always be the tumor itself. Many things can also evoke or invoke this mechanism. Yes, I'm calling it from the side of the tumor, from an oncologic point of view. So what is important is it secretes inhibitory cytokines, so nothing gets attracted to it. It inhibits, it induces apoptosis in those cells which they are supposed to process the antigen and present to the T lymphocyte. It, of course, we already know about the immune checkpoint inhibition, which is provided so that the antigens on the surface of the tumor cell are not recognized by the T lymphocyte. So if you have if you can reinvigorate the immune checkpoint blockade, then of course you can overcome any of these types of blockade suppressive mechanism which is induced by a tumor. The tumor microenvironment, as I said, can of course be mediated either through contact, for contact, that is contact dependent suppressive mechanism, which is very true for the PD1 PDL1 reaction, and it can be independent of that as well. So if you have lot of cytokines which are elaborated by the tumor, they will circulate all over the body, not just in the tumor microenvironment, and that can prevent anything from detecting that there is a tumor there and attacking it. So that means if you can uh, use the security guards of the body to form a palisade against a fence around you, then you will be very easy for it is becomes very easy for you to prevent the antigen presenting cell from reaching you and processing your antigens and presenting it to a T effector cell. So the T regulatory cell is actually the kingpin we feel in the tumor masquerading here. So we can also do how to do an anti-tumor therapy, Treg targeting anti-tumor therapy. Well, these are hypotheses, but already been tested in the lab. We are yet to see them in the clinic. They are in phase 1 to phase 3 trials. So you can do a Treg depletion in the tumor microenvironment. We know we can have a kinase inhibitor. We have an anti-CD25 monoclonal antibody. We can use a simple drug called cyclophosphamide. The kinase inhibitor of interest for many people is what is used in hepatocellular carcinoma. So you can use the kinase inhibitors which we routinely use in hepatocellular carcinoma to deplete a tumor of Treg cells, and that then the patient's own effector T cells can take over, or you can halt their migration. So you can use CCR4 or anti-CCR8 monoclonal antibodies or small effector molecules to do that, or sensitize intratumoral Tregs to a checkpoint blockade. So we already have in the lab, we need to synthesize them and see whether going to be effective in the clinic. So TIGIT, anti-TIGIT and anti-LAG3 also are available. We can also use targeting co-stimulation on Tregs by using a large number of antagonists. We can use Treg cytokine secretion, targeting Treg cytokine secretion, block the cytokines released by the Treg, which are preventing effector cells from reaching the tumor. And of course, Tregs, as I said, are pretty fragile and you can attack them by many other means. One simple mechanism of attacking the Tregs metabolism appears to be metformin. There are now very well characterized large studies done from the MD Anderson Cancer Center, not in their country but in Brazil, of young women with breast cancer who have been given metformin along with their standard chemotherapy drug and were continued on a very small dose of metformin, 250 milligrams. Nobody will become hypoglycemic, don't worry about that, every day for the next couple of years. And it's a randomized double-blind trial. There was a placebo of metformin also given to the other. It ran to about 40,000 women on this side and 40,000 women on that side. And we know the relapse-free interval or the overall survival in women given metformin is pretty high. And metformin seems to do that by altering the Treg metabolism. So you remove Treg from the tumor microenvironment, you can allow the effector cells to attack your tumor. So I have listed here what the thymus actually elaborates. I should have shown this a little earlier, but anyway, how it controls how it inhibits activity of T lymphocytes, or how it promotes the activity of T lymphocytes. A huge number of cytokines are secreted. We can use Treg therapies in two ways. One, let the Tregs remain in the body, and let us give a combination of drugs into the body and overcome the Treg inhibition for the effectors in the treatment as a treatment. That seems to be a little too difficult to do. So that is, but it has been used. First generation approaches were allergen immunotherapy and low dose interleukin 2. And in fact, the first treatment which worked in malignant melanoma, disseminated malignant melanoma, is low dose interleukin 2. That seems to have actually paved the way for most of the immune mediated approaches against cancer. What we need to understand is it's not going to be very specific for the tumor alone. Whatever type of drugs which we are giving to the patient, it will work at other locations as well. So it lacks specificity, and ensuring compliance of daily dosing or twice daily dosing or thrice daily dosing is difficult, and the results are a little unpredictable. So we need to have some modification of the delivery of interleukin 2. If you give a higher dose of interleukin 2, as you know, has to be given intravenously as a continuous infusion. Maybe we can make a nanomedicine out of it and give it, or we can target Tregs itself. Well, what can we do outside the body? Yes, outside the body, we can definitely expand the Tregs. One, so you take a donor specific Tregs, and then you can make them specifically manufacture to attack the, to remove the obstacles from for the attack on the tumor. It's not going to attack any tumor, but it removes the obstacle so that it becomes highly specific. So we already know there are some antigen specific, we call them CAR T cells or Treg CARs, doesn't matter. But at the moment, the CARs which we manufacture are not specific. They don't contain any Treg alteration. So we don't know. We are just targeting, removing all the T lymphocytes. We don't know what are the subgroups which we are removing, inserting the CAR into it and sending it back. The insert, of course, is against the epitope on the leukemic cell or the non-Hodgkin's lymphoma cell or the multiple myeloma cell. Typically, we will target CD19 in a non-Hodgkin's lymphoma and the BCMA in multiple myeloma. So you have wonderful optimized suppression when you manufacture a CAR. Can we do that for a Treg? We are not there yet, because the number of Tregs which will circulate even if I collect about 8 liters, purify about 8 liters of blood, I don't know how many Tregs I am going to get. So I will just end with the few slide, a small slide here, which Sugunakar shared with me. Mary Brinkley never appeared in the Stanford rankings. Last a couple of weeks back, everybody's WhatsApp or social media is full of Stanford rankings. Are you in the top 2% of the world's scientists? No, she was never. She just published 34 papers, not millions and hundreds. She did not chase any citation, no metric, never was in the spotlight. So, what truly matters in science is the question. She never ran behind any list. She was always running behind truth alone. In the end, it's not about how many papers you publish, it's about how deeply your idea can reshape the world. Focus on your idea and not your ranking. I think last slide, I think, challenge ahead is their Tregs are not the common cells which occur in the blood. They are a very small portion of T lymphocytes. And during isolation, the anti-inflammatory Tregs are often contaminated with pro-inflammatory cells. So isolation becomes a problem. Unfortunately, if you take it from a tumor environment and culture it, the proclivity and the homing ability is lost. That means I culture the Treg outside the body, expand them, and then infuse them inside. They don't seem to go anywhere. They are very rapidly cleared from the body. Even if you infuse it in millions, the body has a tendency to rapidly control their numbers. We are not very sure how it controls these numbers. Is there a ratio which it should keep of CD4 cells to CD4 plus CD25 plus positive cells? It looks like, yes, there seems to be a negative feedback loop from there. So without lowering the CD4 count, it at the moment does not appear to be possible to allow the body to retain the infused cells which you have manipulated ex vivo. So the challenge therefore is to sustain what we call a pharmacological dose of Tregs and modify them to home in onto a site of disease. This is the important part. So thank you for your patient hearing. I will end here. There are many more slides, but may not be relevant for this audience at this moment. Okay. Thank you very much for your patient hearing. I will stop sharing. Okay. Right.
Thank you so much, sir. It was really wonderful session, and the way you narrated the story behind the Nobel discovery, I mean the battle and the balance, and explained those the complex concepts in a very simple and engaging way, and it was very easy to follow. We are really grateful for the clarity, the depth, and the inspiration that you brought to the talk today. And thank you once again, sir, for your time and sharing the insights with us. And it was very short of time, and we asked you immediately said, "Okay, let's go ahead." Thank you for that, sir.
Thank you. Thank
Thank
you.
you, sir.
Thank you for the opportunity.
Thank you, sir, for your. And we'll have our next invited speaker, Dr. Prabhala Rao, sir, on the floor, and after which we'll be following with the Q&A session. So we request you to, stay with us for, a couple of minutes so that we'll go ahead with these things. And now let us, it's our honor and privilege to introduce our invited speaker, Dr. Prabhala Rao sir, from the Dana-Farber Cancer Institute and also from Harvard Medical School. So he is a leading expert in cancer immunology, widely recognized for his pioneering research in dysfunctional Treg cells in multiple myeloma. And he have also published a paper on that, opening a new pathways for targeted immunotherapy. And we are delighted to have you with us today, sir. And now the floor is open to you.
You are on mute, sir. Please unmute.
Can you hear me?
Yes, sir. Yes, sir.
Yes.
Thank you very much. It's an excellent talk. I just want to emphasize what he emphasized in the talk is that how they challenge themselves to identify, and Nobel Committee goes after the initial observations rather than T regulatory cells studied by number of people, Ziegler, Shevak, Rodensky. The number of people studied and published high quality papers, but they went to the root observations. So that tells you, like as he said, how many papers you publish, where you publish is irrelevant. Your discovery will shine, and so that is very important. And some of the things are T regulatory cells are activated by number of things, estrogens, and vitamin A, and number of cancer drugs will activate, and the predominantly couple of cytokines such as IL-10 and TGF beta are very important for Treg maintenance. That was the main reason, as the previous speaker mentioned, that T regulatory cells are accumulated at the tumor, and particularly solid tumors, because of the microenvironment that they create with lots of TGF beta and IL-10. Most of the tumors produce these immunosuppressive cytokines. So, and also we studied number of subsets within the regulatory T cells. The reason also is that tumor itself is a self-antigen. No matter whether it is mutated or highly producing high quantities of given like a KRAS and other things, but tumor cell itself is a self. So Tregs will protect in that regard. That also primary reason why they go to tumors, because they're trying to protect, because they were educated. Tregs act sometimes non-specifically, sometimes extremely specific, antigen specific in nature. So T regulatory cells, and my first paper published in 2000. And because myeloma has a hyperactivity and T regulatory cells are not functional. That is our observation. That has something to do how the myeloma does. So I will stop there. If somebody has.
Sir, I'm gonna mute everybody and unmute you, because there's some people who are turning on the
Right.
yeah.
Reason. So I will stop there, and if anybody have questions, more than happy to answer.
Yes, sir. Thank you so much. And the floor is now open for questions, and let's keep this engaging and the exchange of ideas. So, if any of the participants have questions, you can raise your hands, you can react the emojis down there, or you can hit the chat box, or you can unmute yourself and post your questions.
Hello, this is Kiranmai from University of Washington. I have a question to Dr. Raghunadharao, sir. The question is, when are these T-Rex produced? Because in the first experiment of Sakaguchi, you said that the athymic mice are autoimmune, but when it comes to human disorder, IPEX syndrome, it was in the fetus that it was attacked, right? So I just want to know that at what stage of development these are crucial.
It appears that at birth, at least you need a functional thymus to recognize self and non-self. So if you don't have a thymus at birth, you don't have any immunity whatsoever. So you can be attacked by anything and everything, from including your gut microbes. They can wander all around the sun. That means you will require immune privileged location like a bubble to stay in and not be attacked and killed. So you need a sterile environment if you want to survive without a thymus. So anything and everything will grow spontaneously without your thymus. So at least at birth, for a few days, you seem to need the thymus. So the thymus actually, in this experiment, he felt that at least 3 days of life for the mouse was important. I don't know in a human being whether we have data. We do have adult patients in whom we routinely remove the thymus. We had a myasthenia clinic in Nizam's Institute. We removed probably 400 to 500 thymus. But myasthenia, as you know, is itself an autoimmune disorder. So if you remove the thymus, you see the myasthenia abates. But the myasthenia abates only if there is a tumor in the thymus. If there is no tumor in the thymus, it doesn't seem to abate. So it looks like self and non-self is a learned behavior, and you require the thymus for some time after the birth to learn, finished learning. So if you assume that by adolescence, the thymus atrophies, beyond 14 to 16 years, you don't find a thymus. There is a remnant of a thymus. In adult autopsies, in fact, you just have some fibrous tissue there. There is no thymus at all. So because T cells survive in the body very long, they can survive as memory cells permanently in the body. Of course, the number of cells per putative invader becomes small, but the repertoire can rapidly enlarge when there is a re-threatening. That means a repeat threat is there, it can enlarge. So at least in the mice, at least for the first few days, you need the thymus. In human beings, we presume that the adenoid, the appendix, the thymus all serve a similar purpose, like the Peyer's patches in the intestine. They are not just lymphoid follicles left there. They are trying to educate the T lymphocytes into recognizing what is self, what is not self, what is a pathogen, what is not a pathogen, what is a commensal, what is not a commensal. All this is an educational process only.
Thank you, sir. Thanks for simplifying us. I was so delighted to see you here in this platform. And my hi to Madhuri ma'am. She is from my previous PhD institute. Thank you all.
Yeah, thank you. Sir, we have one question in the DM that is from Rupanidhi Srirama sir. He was asked that one query is how does a T-Rex cell kills a rock or slip the T cell?
You're muted, Dr. Rao.
Yeah, I saw that.
Yeah. Sir, my doubt is if any helper T cells or killer T cell or what is it called, the regulatory cells, if they want to kill me, they should have a corresponding receptor. Earlier, we used to say MSC class 1 and all those things. Here, what is that?
Okay. The MHC class 1 is required.
1 minute, sir. What is the slipper T cell missing so that T regulator cell will identify the T slipper cells?
Okay. The killer T cells don't require any identification whatsoever.
Yeah.
So they will kill indiscriminately wherever they are activated. Number one. Two, if you want a T cell to recognize, two things need to happen. There must be an antigen processing by a macrophage, a monocyte, or a dendritic cell. Typically, it's the dendritic cell which processes the tumor antigen and then present it next to the adjacent to the major histocompatibility complex. Only when it is presented with the major histocompatibility complex can it recognize. If it is not presented next to the major histocompatibility complex, it is not recognized at all. So tumor cells are very smart at down-regulating the major histocompatibility MHC expression on their self. So most tumor cells like spermatozoa or syncytiotrophoblast don't express the major histocompatibility complex. So even if they express the antigen, it is not recognized by the dendritic cell. So it cannot be processed. So
yes,
that is how they agree.
Yes, sir. My interest is how the slipper cells attack the normal cells so that autoimmunity will develop. Okay, so in this process, T regulatory cells are killing the slipper cells so that autoimmunity is reduced.
Yes, you're right. So what it is doing is if there is an autoimmune phenomenon by the T effector cell in the body, the T regulatory cell needs to control the effector cell from killing. So the killing happens if there is a disruption of the cell membrane beyond the fetal life. This is what the assumption is. So if there is any surface epithelial change,
receptors and ligands have to be identified, sir.
You are right, sir.
Ligands have to be
identified. You are absolutely right, sir. Yes. If there is any difference, the autoimmune phenomenon seems to be triggered through a viral infection. Typically, it is assumed like a viral infection. At least in acute rheumatic fever, where the valve of the heart as well as the glomerular basement membrane are attacked, we know that it is the streptococcal antigen which is actually coating the surface of the glomerular basement membrane as it is filtered through, and the surface of the acellular heart valves. Heart valves are like cartilage, they don't carry any blood vessels. So it is coated. When it is coated, the surface is altered, and that is the trigger for you to attack. So there is an alteration even in the autoimmune system. It is not as if there is no alteration, but the so-called self non-self class is supposed to be finished as soon as you come out of the mother's womb, and in the next couple of days, at least in the mice, maybe a decade in human beings or shorter. We don't know when it actually ends.
So with this T regulatory cells now, shall we cure SLE, sir?
Well, you can use the T regulatory cell to prevent the T effector cell from attacking the its own body cells. That is the idea. You're right, sir.
Thank you,
sir. Okay. Yeah, go ahead. When a Nobel Prize was presented, this is Dr. Deepak speaking, when Nobel Prize was presented, it was said very much that this is a basic research which they are trying to identify as an change in the field. But there are approximately 200 clinical trials which is presently running on to regulatory cells, and maybe they are going to come into practice soon. So my question was to Raghu sir in a similar manner that as per his knowledge, what all clinical trials are presently running in the field where T regulatory cells are involved, and in addition to autoimmune, is there any chance that this can be used in cancer treatment also, maybe in case of solid cancer? To Raghu sir. Thank you.
With most of the trials are in phase 1 to phase 2, both in autoimmunity as well as in cancers. Majority of the trials are in phase 1 and phase 2. As you said, there are about 200 trials actually running. My feeling is we need to figure out how to sustain the T regulatory cell from slipping back to the 2% norm. So if you allow it to slip back to the 2% norm, then it won't make a difference in any disease. So you need to allow it to sustain at a pharmacological dose. So the entire exercise now is to see how you can sustain the T regulatory cells long enough to be present in the body to remove the camouflage by the cancer or prevent the T effector cells from attacking its own body tissues. So in fact, we want to induce an autoimmunity. Yes, possibly. We can induce an autoimmunity by using simple cyclosporin. You don't require very special drugs. I can remove a simple sample, 10 ml of my own blood, do a fecal hypec, take the Buffy coat, add a little cyclosporin, incubate it for a couple of hours and return it to my body. They will not behave like the original, original T cells. They will now behave as if they have some autoimmune disorder features. They will start attacking everything in the body. Yes, so yes, it is possible, but how to sustain this in the long run is the problem.
Yeah, thank you, sir.
Yes, sir. And then we have a question from Dr. Shivaji.
What
So Dr.
key
Shivaji,
experimental evidence convinced that FOXP3 is the master regulator of T cells rather than a secondary marker of activation? He actually did a series of experiments which I showed you, which typically tell you that if there is a dysregulation of FOXP3, you take the IPEX itself, the syndrome of IPEX, which is easy to study because it's X-linked disorder, or the Manhattan Project's mice. Yes, it is possible to say FOXP3 is the master regulator of Tregs. Why do you feel that it could be a secondary marker of activation? It doesn't look like it is a marker of activation. Well, you may be right to a certain extent. It is quite possible that all the Tregs start off as CD4 positive cells only. They earn the second badge as regulator after a certain activation to produce CD25. Yes, you are right. That way, you are absolutely right that it's a marker of activation. So, if a ordinary T effector cell or a CD4 positive cell acquires a CD25, then of course it becomes a T regulatory cell. And we know that if that is controlled by the FOXP3, the expression is controlled by FOXP3. Both the experimenters, both the groups from across the Pacific showed that. So that means you may be right in saying it's a marker of activation, but it's a marker of activation from CD4 to CD4 plus CD25. Yes, you are right in that way. So both ways it meets the criterion. I have one for Madhuri Subbaya from NAYAB. Are there any studies or infusion Tregs or drugs to induce Tregs for treating autoimmune disorders like rheumatoid arthritis? Yes, of course. We use the same soup as we use for organ transplantation in autoimmune disorders, and all of them work. And they seem to work through Tregs only. So they seem to increase Tregs in the body, and that increase in Tregs through which can be sustained because you are giving the drug on a daily basis seems to abate the disease. For example, you can give cyclophosphamide, you can give methotrexate, you can increase the Tregs. There is no problem whatsoever. So yes, we use those drugs. We use a large number of drugs which we use in transplantation immunology in autoimmune disorders also, and it works. Do you think Treg FOXP3 discovery closes the chapter on immune tolerance mechanism, or just beginning to uncover the deeper layers of regulation? No, they never said it closed the chapter. The chapter was closed on them, and they opened it and said it goes beyond what was accepted in 1980, that it is central immune tolerance and not peripheral immune tolerance. So we now have a whole chapter of peripheral immune tolerance. As adults, we don't have any thymus whatsoever. The only way we can have any more thymus is if you develop a thymic carcinoma or a thymic lymphoma, or like a cricket player, you can get a germ cell tumor in the region of the thymus. The only three tumors which can typically occur there. But otherwise, yes, of course, whether there's a deeper layer of regulation, we don't know. Whether there is something deeper to study. At the moment, it looks like the FOXP3 discovery opens, it doesn't close. The chapter on immune tolerance, how to make it the body tolerate its own self and avoid an autoimmune disorder, and how to uncover a tumor covering itself or surrounding itself by the body's own security guards.
Okay. Hema, I had a question. If there is time for questions.
Yes, sir. Yes, sir. We still have time. And there is
Okay.
one question. You can proceed for the question, sir.
Yeah, sir, thank you very much for a fantastic talk. My question was regarding, regarding some of the drugs that are used.
Yeah, cyclosporin we can
for-
use. We can use sirolimus, everolimus. All of them stimulate Treg only.
Okay, okay.
So
And,
when we use them in hepatocellular carcinoma, both in actual human patients and in vitro, in a model, we can demonstrate Treg upregulation.
Okay, okay. Yeah. So is there any way, to look at patients? You are a clinician, you must be seeing so many of these cases. You talked about, breast cancer and, some of the treatments and all these things. Is there any way you can do perhaps some omics studies and say who is, who will benefit from some of these drug treatments?
Sir, I may not be the right person to give that answer. Possibly, yes, you are right. Who will benefit from a simple tablet like metformin, which is ultra cheap, probably costs about 25 naya paisa a tablet, and practically without any side effects. We know for a fact that metformin, which was discarded by the West but was used extensively by India. In fact, it was a drug which was totally discarded in the United States, has staged a comeback because of its ability to prevent recurrences in cancer. We know that it alters the proteomics of a cell. There are many studies on how metformin alters the metabolomics of a cell and proteomics of a cell, but I am not an expert in that. I did read it some time back when I had to evaluate a paper for a project from Conquer Cancer Foundation. That's how I knew a little bit about metformin in breast cancer.
Right, sir. The reason I'm asking is, now the technology has moved so fast. If there is some way of, particularly for metformin, as you rightly said, it is it's so much affordable and people can use it and it's very, very safe. If there's some way of stratifying patients, looking at their
I think somebody needs to take up. There are so many scientists listening in on to this topic today. I think they can take it up.
Sure. Yeah, thank you. Yeah, back to you, Hema. Thank you.
Yes, sir. Hopingly, we hope that scientists will give us the solution.
Yeah.
And participants did. Yes, sir.
One item. See, these autoimmune diseases in the children, they appear, but as they grow up, so they disappear. In the children.
IPEX, they don't live, sir. They will die.
Yeah.
IPEX children don't live.
Yeah,
the other children, for example, exogenous allergies. I will share you a very simple example. Most of you are very familiar with the way Indian mothers bathe their children. They use basin powder to wash the child because it is a very mild alkali. Saponin. Basically, we are using the saponin from the grain. We are not using any detergent. Okay. But if you have a child who is reared in the United States, your own family member, and comes to India and you use the saponin on that child, they develop intense allergy.
Okay.
We are not sure why that happens. If the child were born in India and was from the earliest days of the life, they are exposed to the same saponin, they seem to develop a tolerance. They don't develop the allergy.
Okay.
But after the first few months or first few years are passed, and you try to expose them to a plant saponin, they develop an allergy. That's very interesting.
See, something like pediatric nephropathy.
Yeah.
But
yes,
it goes off as they grow.
Yes, nephrotic syndromes, yes. Sometimes they will go away, sometimes they don't. Depends upon what is the etiological agent. Minimal change glomerulonephritis often responds, remits spontaneously. The initial days only, first couple of months you need to support the child when they are in the anuric phase. But after that, you don't have to worry at all. They become normal adults.
They put on all steroids.
Yeah. Mostly now steroid with something to spare the steroid.
Okay. So GBS, Guillain-Barré syndrome.
Guillain-Barré syndrome, there is excess of antigen which we need to mop up. So we use a polyclonal immunoglobulin. Sometimes we use rituximab to mop up the excessive antigen. Or you can do if your diagnosis is pretty early, you can do a plasmapheresis. So therapeutic plasmapheresis, where we remove the plasma, 8 to 10 liters of plasma, and supplement it with plasma from the blood bank. That also works dramatically well in Guillain-Barré. So there is excessive antigenic stimulus there, and the body is trying to destroy anything which is coated with that antigen.
Yeah. It takes sometimes, it takes several months to come back to the.
Oh yes, sir. Yes, it comes back. In fact, when we use immune checkpoint inhibitors against PD-L1 and PD-1, we produce a variety of immunological disorders. Every single organ, hypophysitis, cerebritis, cerebellitis, medullitis, pneumonitis, carditis, gastropathy, then colitis, nephropathy, neuropathy, you name it, we can injure any system in the body. And all of it is immune mediated. That is the tragedy of the whole thing.
Thank you very much.
Yes, sir. Thank you, sir. Do we have any other questions, participants? One question we can take, one final question. Considering no.
If not, let us invite Dr. Ajit to give the concluding remarks.
Yes, sir.
Sure. Yeah, thank you. Thank you, Emma, and thank you, Professor Edana.
Yes, sir.
Professor Digumarti, it's fantastic, to hear your talk, in particular, clinician giving a talk in the manner that you have given. I think it is very good because whatever you're talking is all, very much applied, and I think all of us greatly benefited from your insights. I think in this particular case, the Nobel Prize was given to understand how immune system works and how it is, how it is put on check, and the role of FOXP, which I think all of us have heard a lot, but it's nice to see teams working across the world trying to understand this and then, sharing the Nobel Prize. In addition, I think your, analysis of the data and then talk about Treg and the role in autoimmune systems. I think all these are fantastic because these are you're talking from your own experience, which makes huge difference for all the participants here to hear from you as to how the discovery was done and how it is applied and all these things. That's on the science side, and Dr. Raghunadharao.
Yes,
on the learning point of view for all the scientists here, including us, it's a question of what you have to aim for. I think you emphasized that very well. The focus has to be, the final goal and not on the publications and all these things. Number of papers, visibility, all those things, matter only to a small extent. But if you have a goal of identifying some, very, very important fundamental, quest to identify what the problem and how you sort it out, or perhaps over decades. And this is, after 25 years is the one that they got the Nobel Prize. Okay, so that actually emphasized very well. Again, thank you very much. This reminds me of when Sanger got the Nobel Prize. Okay, I think he had about 30 papers, and he got
Yes,
two
sir.
Nobel
Yes.
Prizes.
Yes.
Right.
Yes.
He's a simple. I think it was a PNS paper. I remember this was in the 80s.
Yes, sir.
He sequenced 5x174. Nucleic acid sequencing. And that was a PNS paper, and that gave him, perhaps after that he would have published a few more papers, but that gave him the Nobel Prize. So I think you emphasized the point very well that we need to focus on the real problem and try to put our heart and soul into it and then work towards that rather than worrying about publications and visibility and all. So thank you very much, Dr. Raghunadharao. Okay.
Thank you, sir. Thank you for those kind
Thank you.
words.
Yeah, thank you very much.
And really, on behalf of all of us from the FABA team, we thank you, sir, Raghunadharao, sir, for sharing your time, wisdom, and all your passion with us today.
My sincere thanks to Professor Redana. I was already actually reading it.
Yeah, okay.
I made a few more slides, but they were for my own students.
Okay.
So I thought to make this talk interesting, if you tell it like a story, it will be more interesting.
Yeah.
Professor Redana actually shared a lot of papers today.
Yeah.
I added a few more slides from there.
Yeah.
You made it as a very nice story, sir. That is good.
Thank you.
Yes, sir.
Thank you,
It was
sir.
really very engaging. And it was an absolute delight listening to you. And the participants also can hit the emojis down there and you can share your thoughts and your reactions also. And we also thank Prabhala Rao sir, for coming up with his thoughts, and Ajit Kamath sir for concluding these things, and all the participants. And I'm sure that everyone here is going to leave this session with a little more curiosity and inspiration. And thank you everyone for joining us this Friday, and stay tuned for next Friday. I hope we'll be going on in series every weekend and we'll have more engaging sessions.
Thank you. Bye-bye.
Very nice. Thank you. Thank you, Hema.
everyone.
Thank you.
Good night.